Well, first, I would like to thank the organizing committee for the invitation to come to the symposium and to talk about my research activity for the last few years. So, the outline mainly related to the carbon nanomaterials. First, history, very brief history on nanotechnology and nanomaterial. And as you can see, the first person who was talking about possibility to manipulate atoms was Robert Feynman in 1959. And that was followed by the term itself, nanotechnology, was used by Norio Taniguchi in 1974, and Alex Drexler also was saying that the new area at present in 1986 that this coming area was related to nanotechnology. And two inventions in 1980 with two scientists with Binning and also Hoover about the scanning tunneling microscope and atomic force microscope. And from the 1980s to and 2000, the number of papers that has been published on nanotechnology increased extensively. So saying that, let's say the example, one example of this manipulation of atom on the next slide. This is the slide with HOPG, for example, as the surface highly oriented pyrolytic graphite, and we can see each carbon atom and the radius of the carbon atom and the distance between carbon atoms is correct by STM that we have done in our equipment. And second point, we use the Lamy-Blodgett film of chlorophyll A, two layers, two chlorophyll A, and we see the two layers in terms of thickness here, and in the term of the presentation, the morphology. So H square corresponds one chlorophyll A molecule, because we have for each chlorophyll A we have 26 pi electrons. That's the reason why that we have a good electrostatic, I would say, and also electric current that can be involved with the chlorophyll A. Well, I want to mention that the one that has been a lot of work on this system, mainly for Lamy-Blodgett films, is Dr. Tazi, who is a student who came to my group from Morocco, and he has done a huge potential of work, beside mainly on the preparation of the equipment. So, what is about the classification on the different size of carbon dioxide? The most important, of course, is the one between 1 nanometer to 100 nanometer. And we have this panoply here, the different system that has been worked out for the last at least 60 years and still is used in medicine as an example. So what about the classification in terms of nanomaterial? So first of all, based on the dimensionality, so we can see the chlorophyll, sorry, we can see the fullerene and also the nanotube, and followed by the also graphene, for example, graphene oxide and crystalline compound. In terms of classification, in terms of chemical components, so as you can see for the organic that has been used extensively, micelle and liposome. Inorganic, this is quite important because quantum dots now has been, as in inorganic, has been quite, I would say, popular, and in particular for the computer, for the quantum dots computer. And finally, the carbon dots base, including graphene and carbon dots. So they are those two as very important in the term of different application. So this is the main point I want to mention related to nanomaterial from 100 nanometer down to 1 nanometer. So the second characterization as an outline is related to carbon dots in nanotechnology. So in nanotechnology, it has been formed from 1963 to 2004 that was a panoply of different structure. So, for example, multi-wall carbon nanotube and single-wall carbon nanotube, etc. and the other 2 dimension, and and the 0 dimension is related to the carbon dots. So how can we prepare the carbon dots? So, but before that, what is this discovery itself of the carbon dot? And that has been done by Zhu et al. A collaborator, that they have found that on the electrophoresis during the electronic or electric discharge of carbons, talking to possibility to form or to prepare the carbon nanotube. So that's the electrophoresis has shown 2 spots, and he expected that these 2 spots can be related to the impurity in the formation of the carbon nanotube. But Sun took over this work and find out that if you take a solution of these 2 spots and shining light the the solution, and knowing that it has a what I call a filter to have a specific wavelength. So at 450, 500, 560, etc., is a different color for the compounds, for the solution. So the second step was to use a monochromator and to have specific wavelength at 400, 450, 500, and there was a correlation between these two ways to detect the formation in terms of photoluminescent formation of carbon dots. So that was the first, I would say, really, the term itself, carbon dots, has been given by Dr. Sun from Emerson University. So the next slide is related to the different property that is different property, but very, what I call, very important one. Like the small size, the photoluminescent property in terms of quantum yield is very good, and the biocompatibility, this is very important too. So that's the property of our carbon dots. This is the reason why it is so important. How can we get this carbon dots in nature? So if you take honey, for example, and you do the separation of the constituent of the honey, we find out that there is some carbon dots too, with the absorption spectrum as an example, and which is the right region to see the absorption spectrum of carbon dots. Another example is hamburger. If you heat it to 220 degrees centigrade or 300 degrees, so you will get, from the large size, we would see the formation of carbon dots with the right photoluminescent spectrum. Another example, the bread and sugar, and finally the interaction with carbon dots with the eggshells. You have seen the same optical property for the system. This is for the carbon dots in nature. A lot of paper has been published in recent years to show that different plants can give you the right carbon dots too. So what about the lab, to do the same type of work in the lab? So when we is to use the top-down approach or the bottom-up approach. And the top-down approach use the large molecule to make a small one, but using the laser ablation or electrochemical or arc discharge to make it. Another approach is related to bottom-up, so starting with small molecule to the precursor, mix it together, and having possibility to use the, for example, ultrasonic irradiation, the microwave radiation, or hydrothermal, and we produce our carbon dots too. So that's the same way. Both ways we proceed to give the same property that I have just shown for the carbon dots. What happened in our lab? What have we done in our lab that we published last year? This is at least 3 different preparations. For example, the black carbon dots, and using the powder, carbon powder, mixed to sulfuric acid into nitric acid, and we're able to look the optical property like absorption, the normalized luminescence, and also for the detection of the surface of the carbon dots using the FTIR. For the gel-like, so we were able also to make another type of preparation, and using the ethylene diamine and citric acid mixed together. and can use the thermo or microwave spectroscopy to make it with, again, the absorption, the same region as we have seen for the black, and the fluorescence, which is excitation dependent. And we can see for the normalized one, a different excitation, the band move from the blue to the red. And again, we can look by FTR the composition of the surface of our carbon dioxide. Finally, for the yellow carbon dioxide, that's an important one because we use a sonication. So that means we use a methodology in terms of energy, much lower energy compared to the black and also the jet lag. And the correspondence in terms of UV, there is a good similarity with the two others, but not for the uh, fluorescence, because the fluorescent is no more dependent on the excitation wavelength, but we have a specific band related to the yellow carbon dioxide. And this yellow carbon dioxide is really related to phenylenediamine and citric acid mixture. And we use in the case of the yellow, so we are using mainly the we are using mainly uh, the what is it? Well, mainly using the carbon that's prepared with thermo microwave technique. Sorry about that. So two other preparation has been shown. For the red carbon dioxide, we use a derivative of porphyrin, and in that case, we're able to see the luminescence at 700 nanometer. And that's very important because in oncology, we don't have any autofluorescence due to the particle in the blue region. And many of the carbon dioxide that has been prepared with different plants, for example, they present mainly blue emission, and that means not too good for the oncology. And finally, to change the negativity of the carbon that's prepared, because all the carbons that have been shown up to now, they are negatively charged. And to have a positively charged, we need the tetraamine benzene to mix with citric acid, and using the microwave, it was easy to get the formation of the carbon dots. So this is good for the optical properties, but we may ask the question, what is the problem in terms of functionality? If we want to conjugate a carbon dot to a drug, for example, what is the functionality that is involved? And we need, for this case, to use two different techniques. We are talking about the ESCA or the X-ray photoelectron spectroscopy. And as you can see, for what we just showed about the black, the gel, and yellow, as you can see, the content of the carbon, for example, for each of them, and also the oxygen is quite different. For the one with tetraaminobenzene, as you can see, there is not much percentage for the oxygen, and same thing for the nitrogen. So that's very important. What is the meaning when we use an energy that specifically labeled for specific also, I would say, type of moiety that we have in the molecule? When we think about, for example, carbon 1s, oxygen 1s for specific carbon dots. So this is given on the next one. We have the Bohr atomic level with K, L, and M orbital. As you know, the first two electrons will be in the K orbital, and the photon coming from the X-ray photon will kick out the electron from the K level and to go out. That will be ejected. So XPS or ESCA is the same methodology. same denomination. So this is the one that interact with the surface of the carbon dot. There is another technique that we have used. This is the mass spectrum of time of flight analyzer using a beam of, in this particular case, bismuth 5, and we can detect, in that case, different secondary ions from the surface only. And as an example, we have done the experiments last year and published it with the black carbon dust, the carbon nitride dust, the white carbon dust, which is positive one. And if you look at the content, I mean the content corresponds to what we have shown with the previous one, this XPS, the content. So we have the positively charged, we have the negatively charged ion that are escaping from the surface. So this is quite important for the characterization. So next slide would be related to photocatalytic activity of carbon dust, and we'll look at two types, the gel-like carbon dust and carbon nitride dust. So on the next one, we know that if we use a simulation, sunlight simulator for the solar energy, and we look at different size, 2 nanometer, 3 nanometer, and 5 nanometer, we find out that there is, of course, formation of hole and also electrons. And this will produce some specific, I would say, ROS entity to work for the degradation of, for example, the dye. And as an example, the dye that we have done work was related to the rhodamine B, also the nitrophenol, the rhodamine B, methylene blue, methylene orange, and this, of course, is affected by the carbon dots, and we can see the H plus and also the electron will be interacting with these dye. So based on what I've just mentioned, we talk about the gel. We already said citric acid and phenyl can produce the gel formation, and by dialysis we can able to obtain 3 different fractions. And we're very surprised to show that based on the different fraction, that means linked to, as I just said, about the 3, the 5 nanometer, the fraction 3, for example, is the most important one. And the morphology shows that if we take 5 nanometer, roughly 6 nanometer, we have the other one at 3 nanometer, and finally the one at 2 nanometer. This is the most efficient, was related to the 2 nanometer. So there is a size influence on the value related to the photocatalytic activity. So for the different dye I just mentioned, we can see this difference related to the degradation of the dye. The stability of the carbon dots were great because when we use solution of the dye and we mix it, the solution is with carbon dots, so there is a degradation of the, for example, this one is related to AHB. So if we irradiate the solution for 1 hour and we pick a new solution, we can see the same degradation. That means the stability of our carbon dot is great. There is no decomposition of our carbon dot itself. If we do the same experiment with in this particular case a filter, so we can see with the filter, so there is no huge degradation. Without the filter, that's the degradation that we expected. So for the nitrophenol, as you can see, using the fraction 3, we can see the same behavior that we have shown for vitamin B, and that means that with a filter, this degradation is very small. And if we think about the first order reaction for the degradation is also fall, but still we have some degradation. So the next was related to the fact that the use of the scavenger to see if it is a hole is important, if the oxygen, for example, radical is important, and so on. So that's the reason why I use erythia, which is an influence with the hole, and benzoquinone influence with the radical oxygen, and finally IPA, isopropyl alcohol, and the argon when we eliminate. So this is, you can see there is much less, I would say, decrease of the concentration of the vitamin B, for example. So the comparison now for carbon dots, we're using gel and also the one with carbon nitride. So if we can see this B and D for the gel, that again when we use methyl orange compared to methyl blue, so this two is much more, the degradation much more important for the same number of photon absorbed. And same thing for scavenger, IPA, etc. I mean, we can see that there is no scavenger, no degeneration, and with IPA, not much relation to, and finally for DTA and also for the argon, the regeneration is more important. And if we compare with the carbon nitride, the behavior is the same. We don't see much different, except that for the carbon nitride for the whole, and the ADTA is not really involved in it. So the next step is related to the band gap measurement, and but mainly I want to show you the cytotoxicity with C urgent, the gel or the gel or the carbon nitride dots. They are they are non-toxic for the experiments. So the next step would be related to the carbon dots in nanomedicine, and the first example was related to the one for the lymphoma, which is a cancer cells, and cancer cell means when we think about the cell structure, the cancer is mainly related to the nucleus, where the concentration of DNA is very important. So the way to prepare now the carbon does to inhibit the lymphoma is related to, for example, carbon nitride dots, and we add the doxorubicin using the carboxylic group and NH2 group. This is what we call with ADC and NHS. It is a way to have the amidation process. That means to have doxorubicin connected to the carbon dot. And second, what we do is to do the same thing to transferrin, which has a lot of NH2 group for the protein and interacting with carboxylic group. So the system, the most important is this one. And we can see it on the next slide when we look the effect of different for the different cell line. So cell line having cancer. So that means if you look the red one, doxorubicin, the orange is related to carbon nitride dots, carbon nitride doxorubicin for the black one, the green one with the transferrin, and finally the system that we're just talking to, and each of the cell line in terms of inhibition of cancer cell, so we can see that there is a huge inhibition for each of them, and that means that should be good for in terms of in vivo study. The one I'm just talking about is in vitro, and the way to measure it is to look for the in vivo the weight of the mice after 12 days as an example, and we can see that for 12 days the weight is much lower. That means we are able to inhibit the lymphoma in the mice. That's the meaning of it. The next application I would like to show you is related to inhibition of the protein aggregation. So this inhibition is important because there is a link between the Alzheimer's disease with two hypotheses, the tau protein inhibition and amyloid inhibition. So in fact, we're able to use both inhibition to show that we might reduce the Alzheimer's disease using carbon dots conjugated with a specific molecule, cyclone. So the next slide will show you what I have shown for the preparation, not preparation, but how the tau hypothesis is important is the fact that when we look about the neuron itself, that's typically the neuron, and the one that is disease, what we call disease neuron, is the one that tau is no more bind to the microtubule, and that means we have a dissociation of the protein, and that protein means dissociation, that there will be a change in the conformation, mainly due to phosphorylation, and following by aggregation, and we have formation of some on the neuron. So that's important, and we can see that we're using 4 different synthetic compounds. We're able to conjugate each of them to the carbonhydrate dots. And sorry. And as you can see, the functionalization is with the NH2 group. And what is important is the difference in the functionality in each carbon dot. And what we find out is, excuse me, let's find out is the fact that if we look about only with the buffer baseline, which is here, and we have the formation of our tau. If you use carbon nitride with different concentration, with low concentration, so it goes down to a higher concentration, it goes down, and high concentration, so we can eliminate completely the inhibition of, we have the inhibition of the tau. For the S1 I just mentioned, and this is exactly the same behavior that we can see, but much higher, as you can see, much higher compared to the carbon nitride. That's when we have the conjugated of the cycle. And the result means that if you look at the IC50, where we have inhibition, 50% of the inhibition with the different concentration of the cycle. So the one which is gives the most important inhibition is the IC50, and this is related to the fact that we have 2 methoxy group in the molecule that affect the inhibition of the tau protein. So in summary, we have three consequences of what we have shown, that the J carbon dots have a good activity, photocatalytic activity, and also the carbon nitride does. Second point is the carbon dots. Doxorubicin transferrin is a huge phenomenon, huge possibility for the inhibition of lymphoma cancer cell. And finally, we have shown that the cyclone conjugate has a huge effect related to the tau protein and also the amyloid protein. So future experiment correspond to carbon dots on the distribution, so for the neuro behavior assessment. And finally, the work that is working out up to now is to have the COVID-19 treatment with carbon dots. And you have the collaboration, which is quite extensive, very important. Thank you for your attention. If you have any question, I'll be pleased to answer it.